We are honored to be recognized as the National Clinical Research Center for Hematologic Disease, which is certified by our national government, representing the top 3 clinical centers of hematology in China. We have 616 beds in total, perform more than 1,000 transplants each year, and in addition, hundreds of cases with cellular therapies such as CAR-T.
We are focused on exploring new targets of CAR-T for relapsed and refractory AML patients and reduce the toxicity of CAR-T treatment. We have made active exploration in optimizing condition enrichment of allo transplantation, improving transplantation pattern, and reducing the incidence of GvHD, relapse, and mortality post-transplantation. So far, more than 20 clinical trials have been conducted to assess the clinical efficacy of CAR-T therapy targeting various myeloid antigens such as CD33, CD1, CD38, and lymphoid targets such as CD19, CD20, CD22. Over 1,000 patients with hematological malignancy have been treated with CAR-T therapy in our center, including 598 AL patients, 49 AML patients, 236 patients with lymphoma, and 173 patients with multiple myeloma.
During the past decades, our institute has focused on the early diagnosis and treatment strategies of acute GvHD after transplant. We have explored and optimized the classic immunotherapy for GvHD, cytokines and immunocells. Interleukins such as interleukin-35 are initially demonstrated to play a strong immunosuppressive effect through promoting Treg cells that attenuate acute GvHD. Dendritic cell-derived interleukin-27p28 recognize T-cell program in pathogenesis and elevate acute GvHD. Immunocells, NK, gamma delta T, and Treg cells was verified to be involved in the severities of acute GvHD. Currently, we are also trying to modify the Treg cell by screening TCR dominant clones, thus acquiring Treg cells with strong immunosuppressive functions. In addition, we also try efforts to establish the new treatment methods for acute GvHD.
Our group is conducting a randomized, controlled, multi-center clinical trial of azacitidine plus interferon for the prevention of relapse after transplantation in high-risk AML and MDS. Our group has been conducting clinical trials of fecal microbiota transplantation for the treatment of steroid-resistant gastrointestinal GvHD for the past 5 years, and the results are encouraging. Recently, I have been more concerned and interested in the early complications of endothelial origin. Endothelial dysfunction participates in the pathogenesis of various post-transplant complications such as veno-occlusive disease, transplant-associated thrombotic microangiopathy, capillary leak syndrome, and even GvHD. Our group is exploring the use of the constructed scoring system model for early diagnosis and preemption treatment of those early complications of endothelial origin.
Younger adults with newly diagnosed E-line adverse risk AML who receive intensive chemotherapy have a lower complete remission rate and more dismal outcomes than those with AML in favorable and intermediate risk categories. The combination of venetoclax with decitabine or azacitidine had resulted in promising response rates in older patients with AML unsuitable for intensive chemotherapy. Therefore, we conducted a phase 2 study to evaluate the efficacy and safety of venetoclax plus decitabine as first-line induction therapy in younger adults with newly diagnosed inline adverse risk AML. Our study enrolled 42 patients and revealed quite exciting results, demonstrating that venetoclax plus decitabine was an effective therapy that was well tolerated and resulted in low infection rate and very limited toxicity. No deaths in the first month, with a promising composite complete remission rate of 93%. 79% of patients in this study received allogeneic hematopoietic transplantation and an estimated 12 months overall survival of 91%.
As the president of Chinese Society of Hematology, we are dedicated to conduct innovative clinical studies for creating novel drugs and products to finally improve the prognosis of patients with hematological diseases. We have extensive cooperation with many international organizations, including EBMT,